Good afternoon. Again, it's Tuesday, October 13th. This is another hopefully semi-short video to talk about key issues in Unit 1, Chemistry of Life. So in this session, I want to talk about a little bit about monomers and polymers and the process of joining and breaking these things down using two processes called hydrolysis and dehydration synthesis. We touched on this a little bit last year in regular bio, but you lost a good third to a half of the course because we didn't get to finish the class. So anyway, I want to make sure we have a decent understanding. So when we talk about the four major macromolecules, okay, let me again share my screen. We use a whiteboard. Talk about the four major macromolecules. Remember, we have about 116 different elements. Of those 116 elements, about 25 of those elements are used by living things on a regular basis. And of those 25, 6 of them are really critical. And we have the acronym CHNOPS to remember the key 6. And CHNOPS stands for, again, carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Well, these atoms of carbon, hydrogen, oxygen, nitrogen, oxygen, phosphorus, and sulfur, they get arranged into something called monomers. Monomers are small single units that can be built into repeating chains. Oh man, this whiteboard is just not the best. And then monomers, as I said, can be built into repeating chains, which we call polymers. So just think of the word mono as one. So one unit, and polymer is many. That's many units strung together. And the big four are carbohydrates, lipids, proteins, nucleic acids. All of them are built with monomer components that are built into polymers. Now, carbs, you can string the monomer, which are monosaccharide single sugars, together into long chains, big rings. You could have large collections of monomers to make a pretty complex polymer like starch or glycogen or cellulose. Same thing with nucleic acids, DNA, RNA. We're stringing together monomers called nucleotides, and the nucleotides get strung together in very long chains. And as you may already know, the sequence of those nucleotides is what determines our genetic code. And the third one, proteins, are just long chains of amino acids. 20 different amino acids. They work just like the letters in the alphabet. The order you put those amino acids in totally determines what the protein is going to be. And proteins are usually 300 plus monomers strung together. Lipids is the only exception. Lipids you don't really build into long chains. Lipids tend to be sort of short and stubby. They're made of an alcohol called glycerol with typically 3 fatty acids attached to them. Triglyceride you may have heard of. Triglycerides are fats. So an alcohol with 3 fatty acids. So they tend to be a little bit more short and stubby. You can swap out fatty acids, and depending on what 3 fatty acids you attach to the glycerol, that totally determines what the lipid's going to be. But anyway, on a more basic level, monomers and polymers. One of the things that makes you living, makes you different from the chair you're sitting in, makes you different from the table you may be sitting at, is you have the ability to convert monomers into polymers and polymers into monomers. Non-living things cannot build monomers into polymers, which building, by the way, is synthesize. So they cannot synthesize monomers into polymers. And non-living things cannot take polymers and break them back down into monomers, a process we typically call digestion. But you and I can, and it's one of the key things that makes us living. It's our ability to convert polymers into monomers and then take those monomers and build our own polymers that gives the ability to build our bodies, maintain our bodies, and live. So yesterday or today you ate some food. Maybe you ate a chicken sandwich. Well, you just ate chicken, and particularly you ate the chicken's muscle. And the chicken muscle is protein. So when you put that chicken muscle into your mouth and chewed it and swallowed it, it's too complex. It's in polymer form. Your body cannot absorb polymers into the bloodstream and do anything with them. So the very first thing you have to do is you have to digest those polymers back into the original monomers. In this case, you have to digest that chicken protein back into the original 20 types of amino acids. Once you've broken the polymer back into the monomer, the protein back into the amino acid, now you're in business, because in your small intestine, you can move the monomers through the cell membrane out of the small intestine into the bloodstream, and then from the bloodstream into your cells. Your cells then have the machinery to take those amino acids, reassemble them. This is typically done at the ribosome level. Reassemble them into not a chicken protein, but reassemble them into a human protein. That's critical. Our ability to take in a polymer from a different organism, digest it back into the monomers, and then absorb those monomers into our bloodstream, deliver them to our cells, and then our cells can take those same monomers that were just yesterday part of a chicken, and today convert them into human proteins. It's huge. Absolutely critical to life. Absolutely critical to life. This is why we say in 9th grade, you are what you eat. There's nothing original on you. Every piece of you has been used multiple times. On the end of your finger, there may be a protein from a hamburger that you ate 2 months ago that was made from those amino acids. The end of your hair, there may be some amino acids from a protein, such as a fish you ate a month ago. Every molecule on you has come from what you've eaten. You are what you eat. And that is absolutely critical. So life is all about being able to take polymers, turn them into monomers, and then take monomers and turn them into human polymers. And the process repeats. We consume food from plants and animals. We take those plants and animal polymers. We convert them into monomers. The monomers are the universal trade. The monomers then are absorbed into our cells, and our cells have the ability to take those monomers from plants and animals and turn them into human organic molecules. This only happens because every living thing on the planet uses the same building materials. Every living thing on the planet has the same genetic code. Every living thing on the planet uses the same 20 amino acids. It's one of the biggest pieces of evidence that we all have a common origin. They all come from a common source of life, that we are all somehow connected and related. It's this idea that we use the same materials, we use the same codes, we have the same cellular machinery. All of that adds up to a very strong argument for common ancestry. Now, take that a little bit further. Digestion, synthesis, there's more technical processes here. So digestion also can be hydrolysis. Hydrolysis, let me see if I can somehow write that. So hydro, hydro, which means water, lysis, which means to split. Hydrolysis, splitting with water. So you add water to split molecules. Hydrolysis is how we digest molecules. Not to be too gross, but think about a time that you've had diarrhea. That's a heck of a lot of water coming out of you. That's how much water your body uses every day to digest your food, just to give you some context. And then the opposite of hydrolysis is something called dehydration synthesis. I'm not going to try to write that because it'll take me forever. Dehydration synthesis, dehydrate means to remove water. And when you remove water, that is going to open up a new place to bond and allow molecules to join together with a new bond and make or build a molecule. So hydrolysis is digestion, breaking down molecules. Dehydration synthesis is the opposite, removing water and making a bond. Let me see if I can draw a quick example here. All right, so let me take, let me just go into black. So let's take an amino acid. An amino acid has an R group, which you probably know by now. On the one side, it has an amine group, which is NH, oh God, NHH. And then on the other side, it's got a carboxyl group. So I can't make this go any faster. And then there's a carbon down here. Right. Oops. Put that carbon in the wrong spot. Let's get rid of that one. Try that again. So we're going to put that on the central carbon. All right, so that's one amino acid, very poorly drawn. I'm going to draw another amino acid just so we can illustrate this process. Bear with me. I'll try to go a little faster, which means it'll probably get sloppier. Okay, so there's your typical 2 amino acids. So right now I want to join them together to build a dipeptide so I can eventually build a complex protein. And so I'm going to use dehydration synthesis. What's going to happen is this OH group is going to become attracted to one of the hydrogen groups on the neighboring amine group. So the OH group from this carboxyl group is going to become attracted to this H group from the hydrogen group, and they are going to come together in an ionic bond, and they're going to form, I should say a covalent bond, they're going to form water. So we're removing water. Well, when we take out the water here, and we take out the hydrogen here, that opens up a bonding site. Right? If we go through carbon's atomic number 6, it's got 2 electrons on the first level, 4 electrons on the second level. That means carbon needs to make up to 4 bonds. Well, right now, it's got 1 bond, 2, 3, because there's a double bond there. And its 4th bond is open. It's got an open bonding site. There's nothing there. There's nothing there. And over here, the hydrogen left the nitrogen. And so we got an open bond here on this carbon. And nitrogen's atomic number 7, so that's 2 on the first level, 5 on the second level. That means it's got 3 bonds it can make. So it's making 1 bond here, 2 bonds here, but it also has an open bond. So by removing water, we opened up bonding sites, and what's going to happen is this carbon is going to become attracted to this nitrogen. Whoa, that just went haywire. This carbon is going to become attracted to this nitrogen, and it's going to form a bond between the carbon and the nitrogen. And there's what we call our dipeptide bond. It's joining 2 amino acids together in a dipeptide bond, and that's a strong covalent bond. That's dehydration synthesis. The nice thing about it is we can reverse that process. All we got to do is turn around and add water back in. And if we add water back in, it's going to break this bond. And the water is going to split. And when the water splits, it's going to add the OH, whoops. When the water splits, it's going to add the OH group back onto the carboxyl. And it's going to add the hydrogen back onto the nitrogen. And now we're back to 2 individual amino acids. So completely reversible reactions. Take away water, we open up a bonding site, molecules join together. Add water, we disturb the bond, break the bond, and break them back into the monomers. So we can add to what I was talking about earlier. Life is about taking polymers from other organisms, going through hydrolysis, and converting those into monomers that are universally used. Those monomers are at a small enough scale that they can absorb through our intestines into our bloodstream, into our cells. Our cells then, using machinery such as the ribosome, will go through dehydration synthesis, removing water, to build bonds and create more complex molecules into polymers that are human. And that is one of the absolute critical things of being a living organism. So let me sign off with that one.